So now I'm going to be talking about, we're going to focus on safety now in this statistical plan for safety. And what I wanted to start off with is that, I know a lot of the trainees that we've been working with, safety has been a big part of it. And in these early phase studies, a lot of your focus is on safety. And from our point of view, safety actually comes up in a number of documents. So I'm going to talk about two here real quickly. We have a safety monitoring plan that would be written for your study. And this is, in essence, talking about what data is going to be monitored, when it's going to be monitored by who, and how they're going to monitor that, and how they monitor it, and how often they monitor it, that kind of thing. It kind of details all of that kind of information, and you can see just some of the things that are listed here. And then we have this other document that we as statisticians are responsible for, which is our statistical analysis plan. And this is going to lay out really the hardcore criteria by which we're going to say, okay, we really need to be concerned about this, or we could be laying out, okay, if we have a dose escalation study, what are the actual criteria that we're going to set forth to say we can go forward to the next dose or not, and those kinds of things. So the key piece of this is that safety is part of both of these, and we want to make sure that safety message is, really consistent between those documents. So what are we going to look at with that? When we're designing our studies, the first thing we're talking about is our specific aims. And, I want to demonstrate that my drug is better than another. Well, that might be, maybe that's not quite the right thing. Or I want to prove that my drug is safe. Well, that could be really hard because it's hard to prove that it's safe. Usually, especially these early studies, we're really small sample size. We might have, rare events. We're not going to be able to detect that in there. And actually, at the end of the day, maybe the only thing we could possibly say is that, well, no safety concerns were identified. So we need to be very careful with specifying what those safety hypotheses are. Be very clear what we're going to measure and what we want to see. Note that safety is not necessarily an outcome, but we want to focus on the harms and rather than proving safety, like identifying those harms. As we move into the later phases of trials, maybe our focus is not on safety as much, it's more on efficacy, so we're powering it for efficacy, which means we might not have the power to get, identify those safety events. So we've got to kind of balance that right there. And to make this point, to go even further, safety is assessed along the lifespan of this drug. Early on in our phase 1 studies, maybe we are trying to find a dose. So escalating or how we move through doses is really focused on safety. And then we move to phase 2 where we're looking for that hint of efficacy. We are powering for that hint of efficacy maybe, but we're still looking at safety, so we have to still have guidelines and rules for our safety points. And then we move on to phase 3, maybe phase 4. And we're going to expose this treatment perhaps to larger populations and stuff like this, and all of a sudden something comes up that we never anticipated, because it happens. We open it up to stop, just the sample size and that aspect. You're treating more patients, you might start seeing things that you didn't anticipate. So it truly is something that needs to be examined and thought about through the whole lifespan. So how do we do this? How do we come up for a plan? How do we just start thinking about it so that at the end of the day, and here's my proposal, and I've covered this aspect in a thorough way so that whoever's monitoring or making decisions about my study has valid guidelines for looking at this. So I kind of put this into maybe 4 parts, and that's kind of where this talk goes. So first we want to identify what are the potential harms? Can we identify them? Okay, but here's my safety outcome. How am I going to define that? And it depends on how we're collecting things and what we're collecting. And then based on that, what do I expect? And then how much is too much as far as safety? Where do I go for that? And what other statistical questions do I need to think about that are related to that? So what are the harms? I throw in clip art because it's getting later in the day and I still find clip art really funny. So we'll see if you like my sense of humor. We'll see. Okay, so what do we already know? Is this a new product or marketing? What's out there? What do we know about it? Because the more we know about this product, the more it's been used, even though it might be not being used in the population that I'm interested, the more that we have out there, it kind of changes our viewpoint, our perspective on how we need to evaluate the safety part. So if there's a lot of information about the drug itself, then that's great. If there's not much, maybe we're going to have, I think it was Roger, he had a great statement yesterday. There's a lot of information there. You want to focus on maybe particular things. But if this is a drug or device that hasn't been used a lot, you're going to have to cast a bigger net and maybe you are looking at more, or looking for more potential things that go into that. So what do I know about the drug or device, whatever that is? And then we need to have a very clear, full understanding of what's expected due to that versus what's expected due to the disease that you're studying. Because, you know, when we're thinking about things, is this really related to what I'm doing to this patient, or is this just part of the natural episode of things that happen with patients with this certain disease, and you want to tease that out. And then if you expect an event based on some mechanism of action, but you really have no idea what the rate would be, well then that's when we need to go to our, you know, natural history data, or the epidemiology that you might have there to know what it is. Maybe you have a control group that's been, you know, from another study of similar patients, and you can go for that kind of information. But we have to know what to expect. That's a big piece of that. Okay, so we know what to expect with this drug, perhaps, or in this patient population. Now I really want to define the outcome. So when we're talking about safety, where does this come from? Well, it comes from adverse events. And everyone here has had to put together their protocol, so you know the standard definition of adverse events. When we're typically collecting adverse events, this is a passive process. So a patient comes in and says, okay, have you had anything you know, unusual, any medical problems since the last time I saw you? So it's passively collected. And then you record all those, so you have all these adverse events and sent to a data management center. And then we code these things into using MEDDRA terms. And we don't even just code it that way. Then we put it into groups and body systems. And then the way we present it might be this cumulative occurrence of these by treatment group and stuff. And that's what a DSMB might look at, or whoever's, you know, looking at that data. And it's not for you to actually see what the pieces are. But the fact of the matter, when you start grouping things by body system and MEDDRA term and maybe by severity, these numbers get smaller and smaller. And it really becomes harder and harder to see trends in what's going on with that data. And it's a lot of numbers. I know people don't want to see all those numbers. So there are other issues with MEDDRA codes too, because sometimes you might have a single agent that gets or a single event gets reported as multiple symptoms. So how people record adverse events is one of the things we try to really stress when we're training at the beginning of a study, that you don't want to, you know, code the symptoms. You want to say what's the actual event. Sometimes body systems too broad to group things by. Sometimes some terms may code to multiple body systems, so the groups get kind of all mixed up and it's still hard to determine that. So one strategy, rather than say all adverse events or all adverse events related to this or this kind of thing, if you're really interested in a particular type of event that could be related to treating your patient with this particular drug, maybe we want to say anything that falls into this realm, that we're going to consider that a safety event, and we want to collect that on all people. So we're going to form a group of terms and saying this is a safety event that I'm interested in. So beyond the adverse events, which you are going to collect for all, all your patients and stuff like this, I want to focus on these safety events. And instead of being passive with it, every time I'm going to specifically ask for that. Did you have pneumonia? And I can collect it. And so this just talks about forming these groups, but it's more about, okay, I'm forming a group of things that kind of qualify into the safety event, and you want to make them similar and stuff, and really target it to what you're trying to capture as far as those things that are specific for your study and for your drug. And so in doing this, you specifically ask about those events to each patient. It presents it or produces cleaner data and more well-defined safety outcomes for you to then look at in your thing. So we have this, we have what we think, or this is my safety outcome, whether it's going to be all adverse events or this is the safety events. We have what we should expect the rate to be maybe in that, and now we have to say, well, how much is too much? I know what I expect, but how much can I see before I get really concerned that something is going wrong that's not medically acceptable at that point? So you can come up with maybe with a rate that's just not acceptable based on what you expect, or maybe you say, oh, a relative risk of 3 or more, these are some criteria. But this is a kind of place where you start thinking about defining that safety plan based on some criteria. So from that you can come up with perhaps a stopping rule for safety. So for example, if you expect that the rate of symptomatic ICH is going to be 3%, that's what I expect in this patient population. And I know if I start to see anything more than 6%, something needs to happen. So I've set a criteria ahead of time so that as a DSMB, if that's the body that's looking at your safety data over and over, once it gets to that point, then something needs to happen, and I'm specified it that way. Sometimes it's based on perhaps I know this is the rate, and I can use it. It's a certain frequency, and I say, well, my sample size may not be 1,000 at that point because I'm looking at this data all along. So it's more based on this is the rate given that sample size. What's the criteria I can look at? The key part of this is that we need to say it in advance. Developing this safety plan means you're going to think about this, and you think about how much am I willing to accept to continue to move on. You want to say it in advance. You want to have some really guidelines for that. It doesn't mean that you're going to stop. That's necessary, but at least it gives you a place, a criteria to say, perhaps we need to halt and look and see what's going on at that point. Monitor, I say this is a cool thing. Monitoring requires a combination of statistical and clinical insights. Sharon and I were involved in a study. We were the unblinded statisticians for a study, and there were issues with safety events, certain safety events. I guess Chris is not here. Chris was on the DSMB, so he was on the other side of the fence. And we had lots of debates on this going back and forth. The DSMB had lots of debates on this, and then they throw it back at us, and it's like, how do we, do this? And so there is I guess my point is there's a lot of negotiation when it comes to look at these things. Having criteria to say, okay, we're at that point that we need to do something versus, just leaving it up in the air, and then it's just personalities at that point saying, well, I'm not comfortable with this, kind of thing. It becomes really tricky. So it's really cool to have that, and it is that you have to have this dialogue back and forth sometimes to say, is this reasonable or not? Can we go more? Here's an example of what I was talking about a little earlier. So this is just an illustration. Say we know perhaps that we have a study, 2 treatment arms, and we also have 2 age strata for this. And we know how many subjects have been accumulated in each of those strata, but we also know the number of subjects with that particular adverse event, in this case symptomatic ICH. And we can provide numbers, the probability of observing what we're seeing, given that maybe the true rate is 3% versus it's 5%. And we would see that, for that patient in treatment B who's less than 60 years, and we've seen, 2 of the 11 with that event, and the probability that is 0.4, I might be concerned with that. This is very unusual. And the same in the case in total for treatment B, we have 5 out of 51 in the probability of observing that. If the true rate of symptomatic ICH is 3%, is 0.02, I might be concerned with that. And these are the types of tables or illustrations of these things I think that I'm talking about that would be helpful for whoever is monitoring these safety events for your study so they can make that judgment of whether they need to be concerned or not. And here's another one, another illustration that we've used in studies where instead of just having the event, it was important to know how many subjects were observed for that time. So I picked a bad example because I said within 48 hours, but let's just say it was a safety outcome within 30 days, and I'm looking at this data okay, I'm going to freeze the data for the DSMB to look at it. I'm going to have have patients in there that may have only been observed for 1 day or maybe 10 days and not the full 30 days. So I want to, in calculating that rate, I want to take into account how long I've actually observed my patients. And so instead of just looking at that proportion and calculating the rate that way, I would use a relative risk that would actually account for the amount of time that I observed each of the subjects. So it's just another way to illustrate that same concept. Another thing we might look at are, here's a forest plot, where you can see what's expected on the right, that, that if there's no difference, we're right in the middle, but we can look at different AEs and see where they play in that forest plot. So just an illustration of things. Another is a volcano plot, where you can see here on the right, again, what are these particular adverse events that are kind of falling unusual from where we would expect everything else. So it doesn't always have to be numbers by which we're looking at things. It could be illustrations, and this is kind of just useful tools in using what you've already established in your safety plan on how you're going to monitor these things, but in a graphical way that makes it easier for whoever is looking at the data to actually make a judgment. What are the other statistical questions we need to think about? And when it comes to sample, we just talked about who are you using? Are you using your ITT? Are you using, who are you using in this? Well, when we're talking about safety, would it really make sense to include someone who never received the drug? Maybe not, right? So we have to think about that. And so guidelines here, if they didn't get the drug, how can they be harmed by it? So would I really want them in my safety analysis sample? What if someone was randomized, let's just say if you're comparing 2 drugs, A and B, somebody was randomized to A, but they actually got B. Would I want to include them in the group, the A group, for safety because they didn't get that drug? So questions to think about in defining who's included in my safety sample and how I'm going to look at them, how I'm going to treat them in the safety sample. Those are things you want to, you're who, okay? How many? How much do I need for this? Things you have to consider. We in our group have talked about studies where we're looking at 2 group comparisons, or perhaps you're not there yet, where you're randomizing to 1 of 2 groups. Maybe you want to compare, you just have 1 treatment group and you're comparing it to some historical control rate or expected rate. Those things go into the how many question. Whether you want a 1-sided or 2-sided test for that. Am I only trying to say this increases harm, or ultimately, am I interested in both directions or just one? That's another piece of the puzzle. Then how do we look at this? Do we want to look at confidence intervals around an effect size? Maybe we're looking at the frequency events. Maybe we're looking at the time to an event. And depending on that, we might be looking at different statistics, relative risk, odds ratio, or maybe hazard ratios. When I'm doing this, do I want to consider baseline covariates? Is there something important that I need to think about in doing this? All of these kind of go into the how many and how are we going to come up with that ultimate sample size that might be needed. How often do we want to look at this? This still has to be pre-specified, by the way. So we think about, and I can say from a safety perspective, we're definitely really good about efficacy, and if we're in a large trial, we're going to do a group sequential design. We say when we're going to look at it, how often, that kind of thing, because that's all come into play with our sample size. With respect to safety, and from that same study, where Sharon and I were this unblinded statistician, from the very first time we produced a report for the DSMB, there was a safety issue. From the very first time. Very small sample size at that point. And what we thought we would be doing, like producing reports on a 6-month basis, every 6 months, ended up being every month because they were concerned about this safety issue. So although, these kinds of things are important, you want to think about how often you look at it for efficacy, but also how often you would look at certain things for safety. So again, these would go into these plans, so that piece of it. Do we need to adjust for multiple comparisons when it comes to safety? We talked about multiple comparisons yesterday, but we're not trying to prove safety, just disqualify the risk? Do we need to take a hit on that? Maybe we're not so worried about having, inflating our type 1 error when it comes to safety. Because do we want to be cautious or not? Things to think about with this. Again, things you talk with your statistician about when you're designing your study. Oh, and then you have this thing. We're looking at the data. We didn't think about a certain adverse event. I have done my homework and specified in my safety plan, these are my safety outcomes, this is exactly how I want to look at it. And you run the data, and the data are what the data are, and all of a sudden you have these adverse events, these certain things that are popping out that are disproportionate between your treatment group and your placebo group. They happen. It happens. So in that case, like I just said, it may trigger more monitoring of your data. So just know that surprises are not always good surprises. In this case, you wouldn't want that. So why are they found later? We have rare events. We're looking at small sample size a lot of time. Sometimes, it depends on who we're looking at, which patients particularly come in. It just might happen that way. So these things are found, they happen at times. So summary. So the whole thing with this and establishing a safety plan, and again, Roger had a really good saying on this yesterday. We're always balancing the risk and the benefit in this case, right? So we want to have a good perspective about what we should expect. We want to have clear definitions about what's important as far as adverse events or safety events. We want to be able to use that information most effectively, so it might be that we have to group certain things together to say this is what I'm really interested in. And think about what am I, have to establish what is expected versus what is too much before I need to take action. That needs to be established ahead of time. At least it's a good guideline for people who are going to be monitoring the data. You have to be mindful of the sample size and multiple comparisons. We're going to talk a little bit about that. And remember that you can have surprises in the end. They're not always happy surprises, and so it happens. And so anyway, but good preparation is what we always try to do. And that's it. Thanks. Question?
I mean, I don't have to. How much of the plan, like that table for monitoring for the probability that the true rate of ICH is this versus that, how much of that do you come up with and put in the statistical analysis plan, and how much of that is a collaboration, the DSMB asks for something like that?
So with my group, actually, my trainees, I've kind of given them that as a skeleton to kind of, especially those who are doing dose finding studies. So they know, okay, I'm going to look at a group of 20 subjects and assess their safety before I move to the next dose. That kind of table is a very good guideline to say, okay, what would we expect? What's the maximum we would actually tolerate? And then be able to give those kinds of numbers to have an idea. So for reviewers to kind of say, okay, this is a good guide. And you can also calculate with those same kind of numbers, the probability of stopping based on how many number of events you actually observe. So it gives a good indication of that forethought that I've thought about these kinds of things. Not only how far I'm willing to go with that rate of event, but also the probability of actually stopping my study, given I know that this is going to be my cohort, before I would move to the next one, right? So, and then let's just say you funded and the study's running, and we are looking at an interim, and it may not be that full cohort. It's not that we would do it for all, but if we actually have a certain safety event that might be more than we expected, perhaps we want to provide that kind of information. What was the probability of seeing this, given this is what we expected? So it again gives that DSMB some guideline as to say, should I jump on this and do something, or should I just, it's okay, it's within thing. And at least it gives it a statistical framework. The clinical side of that has to